Dear ladies and gentlemen, dear colleagues and friends, it's a great pleasure to see that we actually managed to fill the University Aula. Today we have a fantastic possibility to listen to the Chemistry Nobel Laureate for the year 2012, Professor Robert Lefkowitz from Duke University. My name is Jonas Bergqvist. I'm professor here at Uppsala University in analytical chemistry and neurochemistry, and I'm also chairman of the Chemical Society in Uppsala. Before we will attend the lecture of Professor Lefkowitz, we have, as tradition calls, a fantastic chance to hand out a prize for the best master thesis student in Uppsala during the year 2012. And by that, I would like to ask Professor Lefkowitz to join me on the stage and help me to give out the prize, somewhat less, at least in the money-wise, prize than what Professor Lefkowitz was given in Monday this week. So please, Professor Lefkowitz, could you please help me? And unfortunately, the student of this year's prize is not present. So we managed to get her on a video link. So hopefully this will work.
Hi, my name is Jana Horakova. I'm a student from Prague, and I was doing my master thesis at the Analytical Chemistry department in Uppsala. I'm very honored that I've been selected and nominated for this year's Kemiska Sällskapet i Uppsala Stora Pris for my master thesis work. I would like to thank my supervisor, Konstantin Artemenko, and other colleagues in the lab that helped me with my project work. I'm very sorry I cannot attend this year's lecture and ceremony. I hope you will have a great day, and I wish you all the best. Bye.
So Jana did a fantastic study. She worked together with a colleague of mine in Uppsala, Associate Professor Konstantin Artemenko, and she actually managed to discover protein content on mucosal surfaces in the ovary duct in rabbits. And that's part of a study that are going on in vitro fertilization and fertility problems. So a very important study. And unfortunately, as you heard, Jana is not present, so I will ask Associate Professor Konstantin Artemenko, the supervisor of Jana, to join us on the stage also to accept the prize and the award for Jana's sake. Please, Konstantin.
Thank you very much for giving me the opportunity to accept the prize for Jana, and I was really happy to be her supervisor, and it's a big honor for me to be on the stage with you also and get the prize from you. Thank you so much.
Thank you.
And well, I hope this tradition will continue, and probably we will contribute more in chemistry as you did and have been awarded this year. Thank you very much again.
So, our Nobel Laureates are, of course, a fantastic inspiration for our students and young researchers, and I hope we can continue this tradition and also spread the glorious light of awards and prizes in Uppsala for many more years. I just pulled out, before we are starting the lecture of Professor Lefkowitz, I just pulled out the very nice diploma that Professor Lefkowitz and his colleague, Professor Kobilka was awarded. And as you see, the prize were given to these two gentlemen for the investigation and the development and the scientific evaluation of the Gcoupled protein receptors. And those receptors, for those who are not in chemistry, are fantastic. They are directing everything in life, more or less. And I'm working in neuroscience and neurochemistry, appreciate that, of course. If you look at the living brain and the receptors that are controlling the functions in the brain, these receptors are the key to many of the things we are looking at. And also they are key proteins for instance, drug interaction and when you want to treat patients.
Well, it's certainly a pleasure and an honor for me to stand before you as this year's Nobel Laureate in Chemistry. And I'll be speaking to you today about my favorite research topic, receptors. You know, receptors, the idea of receptors, has fascinated scientists for more than a century. And it has certainly fascinated me and my co-winner, my former student, Brian Kobilka, for the entirety of our research careers. Now today we know that one particular family of receptors, the so-called Gproteincoupled receptors, and they're also known as the 7 transmembrane receptors, represent by far the largest, the most versatile, and the most ubiquitous of the several families of plasma membrane receptors. They comprise almost 1,000 genes in the human genome, and they regulate virtually all known physiological processes in humans. In fact, 3 of our senses, taste, smell, and vision, are mediated via receptors of this family. And in terms of clinical medicine, this is extraordinarily important because more than half of all drugs used in the clinic are drugs which target these receptors either directly or indirectly. Now, despite the very central role that research on these receptors plays in current biomedical research, it's really only in the last 30 or so years that there's even been any agreement that they even exist. Prior to that time, there was a great deal of skepticism and controversy about the idea of receptors. Now, perhaps the earliest fairly direct assertion that that might be something like a receptor can be attributed to the very famous British pharmacologist J.N. Langley. More than 100 years ago, in 1905, he wrote the following: "So we may suppose that in all cells, 2 constituents at least are to be distinguished, a chief substance which is concerned with the chief function of the cell, such as contraction and secretion, and receptive substances which are acted upon by chemical bodies and in certain cases by nervous stimuli. The receptive substance affects or is capable of affecting the metabolism of the chief substance." So here we had, in this very prescient statement, more than a century ago, the idea which incorporated the 2 presumed functions of what a receptor would do: the ability to interact with chemical bodies or nervous stimuli, presumably by binding, and then the ability to activate some cellular mechanism or effector. But this idea of Langley's was not generally accepted. In fact, it was either generally ignored or derided. And an example of this is the following statement ironically, by his student, Sir Henry Dale. Now, Dale won the Nobel Prize subsequently for his studies of cholinergic neurotransmission. But here's what he had to say 40 years later about his mentor's receptor idea. "It is a mere statement of fact to say that the action of adrenaline picks out certain such effector cells and leaves others unaffected. It's a simple deduction that the affected cells have a special affinity of some kind for adrenaline, but I doubt whether the attribution to such cells of adrenaline receptors does more than restate this deduction in another form." So he had very little respect for this idea. Now, even more ironic is this next quotation, which is attributed to Raymond Ahlquist. Raymond Ahlquist was a very noted pharmacology of the mid-20th century, professor of pharmacology in the United States. He won the Lasker Prize for his very seminal paper in 1948, in which he proposed the idea that, based on differing orders of potencies of various adrenaline-like molecules to stimulate different responses, he proposed the idea that there must be 2 different kinds of receptor for adrenaline, which he called alpha and beta. But the idea of receptor to him was really very different than what we think of today, because 25 years later, as late as 1973, he wrote the following: "This would be true if I was so presumptuous as to believe that alpha and beta receptors really did exist. There are those that think so, and even propose to describe their intimate structure. To me, they're an abstract concept conceived to explain observed responses of tissues produced by chemicals of various structure. So, it was really against this background of real obvious skepticism about whether receptors actually existed as any kind of physical entity that those of us who were interested in bringing these mythical receptors to life began the work about 40 years ago. And it was clear to all of us, I think, at the time, that if we were to be successful, it would be necessary to develop a whole suite of new technologies which did not then exist, and that the first of these would have to be some way of directly measuring or studying the receptors, some way of tagging them. And so I set out with students and fellows in the laboratory to develop radioligand binding methods to study the receptors, choosing initially the so-called beta-adrenergic receptor for adrenaline, and then the alpha-adrenergic receptors for adrenaline. And we were successful in developing such methods, and from these methods, we reaped many bounties. We were immediately able to study all manner of factors which regulated the receptors in terms of their numbers and properties. We were able to discover new receptor subtypes. And interestingly, we were able to develop various theories of receptor action. Let me give you an example of one of the more important ones, and it is shown here. We found that in radioligand competition binding assays, antagonists, beta blockers if you will, here shown as alpranolol, gave steep and uniphasic curves. But agonists, such as epinephrine, or in this case the synthetic congener of epinephrine, isoproterenol, gave shallow curves which could be resolved into two components, one of high and one of low affinity. The addition of guanine nucleotides, such as GTP, to these binding incubations shifted the curve to the right and steepened it because it converted all the high affinity binding sites to low affinity binding sites. To explain this surprising behavior, together with fellow André de Leon, we developed the so-called ternary complex model. This proposed that an initial interaction of agonists with free receptor led to a low affinity binding complex, here shown as AR. That then, in a second step, this low affinity complex bound to a second component, shown here as X, to form a high affinity ternary complex, ARX. The ability of guanine nucleotides to modulate these complexes immediately suggested that the X in ARX was likely the then just discovered guanine nucleotide regulatory protein, GS. The ability of an agonist to drive the interaction of the AR low affinity complex with the guanine nucleotide regulatory protein is in fact, of course, a measure of its efficacy or its maximum ability to stimulate the effector adenylate cyclase. In fact, that efficacy is proportional to the ratio of affinities of the agonist for the low AR complex to the high ARX complex. These affinities then, in turn, can be easily deduced from these ligand binding competition curves. And so what we have in the ternary complex model is, and in these data, is perhaps the first direct demonstration of the mutual allosteric regulation of the receptors by the binding of ligand on the one hand and effector, guanine nucleotide regulatory protein, on the other. And it demonstrates the reciprocal ability of the binding of each of these two ligands, agonist and G protein, to reciprocally enhance the binding affinity of the other to the receptor. This formulation turned out to be universally applicable to essentially all members of the GPCR family. But perhaps one of the most important consequences of the ability to tag the receptors with these radioligands is that it gave us for the first time the ability to begin the daunting task of trying to purify and isolate the putative receptors. This was truly a daunting task, and it took my laboratory about a decade to isolate first the beta-adrenergic and then the then 3 other known subtypes of adrenergic receptor. The key to our ultimate success was the development of affinity chromatography matrices, in which we coupled beta and alpha adrenergic antagonists to solid supports. We then solubilized the receptors, a difficult job in its own right, and applied those soluble extracts to these affinity columns so that we got biospecific adsorption of the receptors to the columns, and then biospecific elution using counterligands, other agonists and antagonists. By coupling such biospecific procedures with more conventional chromatographies, we ultimately purified the receptors approximately 100,000-fold from the membranes. As I said, it was daunting work. It took many fellows and students many years to accomplish this. The receptors, in fact, were really little more than trace contaminants of the membranes. On SDS gels, and this slide basically, as I say, represents 10 years of work by many students and fellows. Shown is the isolated receptor proteins for 3 of the 4 subtypes that we had isolated. As you can see, each is a single polypeptide chain, molecular weight about 65,000 Daltons. These were glycoproteins, and each of the isolated proteins bound ligands, agonists and antagonists, with all the specificity and stereospecificity characteristics that one would expect for the appropriate receptor. We thought that this would finally convince people that we had isolated a true receptor. But still some skepticism persisted. Individuals questioned us as to whether we could in fact prove that these isolated molecules could also carry out the companion function of not only binding the ligands, but activating a cellular effector. So to this end, we performed reconstitution experiments. A very gifted postdoc, Rick Serione, in the lab, initially did the following experiment. First, he reconstituted the purified beta receptor proteins into phospholipid vesicles. We then found a cell type which expressed the adenylate cyclase and G protein effector mechanism, as well as other receptors like prostaglandin receptors, but which did not express the beta adrenergic receptor. Hence, the adenylate cyclase of these cells, and the cell, by the way, was the erythrocyte of Xenopus laevis, the erythrocyte of the African clawed toad. Long story as to why we wound up using that cell. Suffice it to say that virtually all mammalian cells express beta adrenergic receptors, but the erythrocyte of Xenopus laevis does not. In consequence, its adenylate cyclase is unresponsive to epinephrine. By taking our reconstituted receptors in the phospholipid vesicles and fusing these vesicles with polyethylene glycol to the Xenopus acceptor cell, we could essentially transfer the receptor protein into the membranes of the cell, which then became responsive to adrenaline. Within a year, and in collaboration with Lutz Birnbaumer and the late Eva Neer, we were able to perform a complete reconstitution of the adrenaline or catecholamine responsive adenylate cyclase system from 3 purified proteins. Remember, nothing had been cloned, nothing was overexpressed. Our purified beta adrenergic receptor, a beta 2 adrenergic receptor, the guanine nucleotide regulatory protein GS, and the catalytic moiety of adenylate cyclase. This successful reconstitution, 1984, proved once and for all that the molecules that we had isolated were truly the beta adrenergic receptor and were capable of carrying out both functions of a receptor, specific ligand binding and activation of the cellular effector. Now, with bona fide validated receptor in hand, we performed experiments with cyanogen bromide. We cleaved our protein. We were able, using then very recently developed microsequencing techniques, to come up with 5 short stretches of amino acid sequence. And based on those, we designed oligonucleotide probes. And after several years of difficult work, and in collaboration with workers at Merck Pharmaceuticals, we were able to clone the gene and cDNA for the beta 2 adrenergic receptor, the first ligand-binding GPCR whose sequence became known. This work in my laboratory was carried out largely by a young cardiology fellow working in the laboratory, Brian Kobilka, who, as you know, has ultimately shared the Nobel Prize with me. And when we obtained the deduced sequence of the receptor, we could see in that sequence all of the features that I'm sure most of you know are canonical for the larger family of GPCRs. 7 apparent hydrophobic stretches, membrane-spanning domains, interconnecting loops, sites for N-link glycosylation toward the amino terminus, and for regulatory phosphorylation toward the C terminus. And shown in blue, what was, for all of us at the time, 25 years ago, a remarkable surprise. Residues which were identical with residues in rhodopsin. Now, rhodopsin, of course, is the visual pigment and was known to be, in a sense, a light sensor. Moreover, it was known at the time that there was a certain functional analogy between the rhodopsin system, which somehow seemed to signal through a G protein transducin, and receptors like the beta-adrenergic receptor, which also signaled through a G protein. But absolutely nobody at the time suspected that there would be any structural relationship between the beta-adrenergic receptor and rhodopsin. Seems so obvious now, but why was that? Well, the sequence of rhodopsin, the complete sequence, had been determined about 2 years before our cloning work. And interestingly, that sequence had been determined not by cloning, but by Edmund degradation, by classical protein sequencing, because one can obtain grams of rhodopsin from, for example, bovine retina. We never had more than 25 to 50 micrograms of purified beta-adrenergic receptor in hand at any one time. Anyway, when rhodopsin was sequenced, it was found to have 7 membrane spans. And at the time, the only other 7 membrane spanning protein known was bacteriorhodopsin. Now, bacteriorhodopsin is a light-sensitive proton pump found in primitive archaebacteria. So, both laboratories which had sequenced rhodopsin came to the same conclusion. Well, we have only 2 proteins which contain 7 membrane spans. Both are light-sensitive, and so the speculation, very reasonably, was that 7 membrane spans must be somehow, and for some reason, a signature feature of light-sensitive proteins. Only with the cloning of the beta-adrenergic receptor did the idea first emerge that, wow, maybe it's really the signature feature of Gproteincoupled receptors. Very quickly, we cloned the alpha-2 adrenergic receptor, again based on protein sequence. And within a few years, we had cloned 8 adrenergic receptors, 3 of these based on protein sequence. Now, thereafter, many laboratories excuse me. Thereafter, many laboratories were able to clone other members of the GPCR family, and so the family grew very rapidly. And ultimately, to almost 1,000 different receptors. But virtually never again did anyone ever have to purify one of these exceedingly rare receptors to get the sequence to clone a receptor. The family expanded by a series of homology techniques based on sequences of receptors that were already known. And so we always felt in retrospect that that very difficult decade that we had spent purifying the first 3 adrenergic receptors had been worthwhile, because the sequences it provided ultimately for these first receptors that we cloned provided, if you will, a Rosetta Stone, which allowed the cloning of the wider family. We next turned our attention to trying to learn how this very unique and seemingly highly conserved 7 transmembrane spanning arrangement of the receptors was specialized to perform these 2 functions of ligand binding and activation of G protein effectors. We used techniques like site-directed mutagenesis and the construction of the first chimeric receptors between the alpha-2 and the beta-2 adrenergic receptors. And again, this fellow Brian Kobilka played a major role in that work. Now, the alpha-2 receptors and the beta-2 receptors, we had learned from their sequences, were very similar. They shared 50% of all of their residues. But both biochemically and physiologically, they perform effectively opposite roles. For example, the alpha-2 receptor inhibits adenylate cyclase through GI. The beta receptor stimulates adenylate cyclase through GS. By making a series of chimeras, we were able to demonstrate that the simple insertion of essentially just the third cytoplasmic loop of the beta-2 into the alpha-2, which had essentially no effect on ligand binding specificity, converted the alpha-2 receptor from GI inhibitory to GS stimulatory. From much other work such as this, in my laboratory and many other laboratories around the world, it became clear that the ligand binding function of the receptors resides essentially in the outer portions of the membrane spans and in the cytoplasm and in the extracellular domains, whereas the coupling function to G proteins and the G protein coupling specificity resides in the cytoplasmic domains, especially those shown in blue here, in closest apposition to the plasma membrane. Now, in the course of performing further mutagenesis studies, Susanna Kotecki, a fellow in my laboratory, serendipitously discovered that mutations right here in this distal part of the third cytoplasmic loop of several of the adrenergic receptors, rather than leading to the expected loss of function, in fact led to the establishment of constitutively active mutant receptors. That means to say that the receptors were active in coupling to the G proteins even in the absence of any ligand. At the time, we conceptualized this as those mutations essentially abrogating certain then unknown intramolecular interactions which normally constrain the receptors to the inactive state, shown here as R, thereby mimicking the conformational changes which would be accomplished when an agonist binds to R, thereby converting it into the activated R star G protein coupled form of the receptor. Brian Kobilka is not here today, but if he was, he would describe how his crystal structures of the receptor have shed light on the nature of those intramolecular interactions or switches which are thrown when ligands bind to the receptor. Now, contemporaneous with this work on the structure of the receptors, I had been very focused in my laboratory on trying to understand the virtually universal phenomenon of receptor desensitization. And that's illustrated here for the beta-adrenergic receptor expressed in a cultured cell line. So when you apply adrenaline or any adrenaline-like molecule to the beta-adrenergic receptors, you stimulate adenylate cyclase, cyclic AMP rises, but then very quickly, over a matter of just a few minutes, the levels fall back almost to normal, even the continued presence of the stimulant. This is receptor desensitization, and it turns out to be an entirely universal phenomenon. I'd always been fascinated, going back to the earliest days of my career, by such desensitization, because it seemed to me the quintessential example of what is perhaps the most pervasive principle of physiology, homeostasis. Living cells have developed myriad mechanisms to oppose almost any perturbation. So whenever you perturb a living cell, stimulate it in any way, it has many mechanisms which tend to oppose those perturbations and bring it back to the basal state. And receptor desensitization appeared to be such a phenomenon. So let me describe how we came to discover what is the major biochemical mechanism of such desensitization. By the way, here's another example for the angiotensin receptor. A completely different receptor, works through a different G protein and a different signaling mechanism. So how did we come to discover the mechanism of this desensitization? Well, about 1980, we had just developed the first photoaffinity probes for the beta-adrenergic receptor. And a fellow in the lab, Jeff Stadell, wanted to apply these to the study of desensitization. So we took cells, desensitized them by exposing them to an adrenaline-like compound, and photoaffinity labeled the desensitized versus the naive receptors. And when he ran such photoaffinity labeled receptors out on SDS gels, he found that the desensitized receptors had a retarded mobility compared to the naive or undesensitized receptors. At the time, it had just been described that phosphorylation of proteins, some proteins, especially membrane proteins, led to just such retardation on SDS gels. So we labeled our cells with inorganic radioactively labeled phosphate, and were able to demonstrate that catecholamine-induced desensitization in our cells we were studying was associated with phosphorylation of the beta receptor. Subsequently, over the next few years, a very talented graduate student, Jeff Benevik, was able to identify the kinase involved in this phosphorylation, to demonstrate that it was a new cyclic AMP-independent kinase, to purify it, to isolate it from bovine brain, and clone its cDNA. And again, by so doing, he established that it was, if you will, the founding member of a new subfamily of kinases. Contemporaneous with our work, and by the way, we named it the beta-adrenergic receptor kinase. Today it's called GRK2 for Gproteincoupled receptor kinase 2. Contemporaneous with this work, workers in the field of visual signaling had found a very analogous phenomenon, because it was known that when light interacted with rhodopsin, it seemed to become desensitized, and this desensitization was found to be associated with a kinase activity, which phosphorylated the rhodopsin, and somehow, again, that seemed to be associated with its loss of function. That kinase was called rhodopsin kinase. We got together with Chris Palchevsky, obtained some purified rhodopsin kinase enzyme, obtained some microsequence from that, and Wolfgang Lorenz in my laboratory was then able to clone the cDNA for rhodopsin kinase, which turned out clearly to be a member of the same new family of kinases as BARC, or GRK2. And so we knew at that point there must be a family of these G-protein-coupled receptor kinases. In fact, today we know there are 7 members of this enzyme subfamily. 2, GRK1, rhodopsin kinase, and GRK7 are found only in the retina. GRKs 2, 3, 5, and 6 are ubiquitously expressed. Thanks to John Tesmer's laboratory, we have crystal structures for a number of these. The first was published 10 years ago in collaboration with my group, and is a structure shown here of GRK2, or BARC, in complex with G-beta gamma, with which the kinase interacts in the cytosol as part of its mechanism for being drawn to the membrane to interact with the receptors. All members of the family have a conserved tripartite domain organization with a central catalytic domain flanked by 2 more divergent regulatory domains. But it turned out that phosphorylation of the receptors by GRKs was not the whole story of desensitization. Turned out something was missing. So Jeff Benevik found in the lab that as he purified BARC, that to our initial distress, it lost its ability to desensitize the isolated receptors, which we were studying in an in vitro reconstituted system. This suggested that the purification might be leading to a loss of some necessary element or cofactor. Then, at just about this time, the late Hermann Kuhn described his finding that an abundant retinal protein, which at the time was known only as 48K protein or S antigen, somehow worked together with rhodopsin kinase to deactivate rhodopsin. And so this protein was renamed arrestin because it somehow seemed to arrest or stop rhodopsin function. This seemed very much like the kind of molecule we might be losing, although we knew it couldn't be this 48K protein, now renamed arrestin, because that protein was known to be limited in its expression only to the retina. Nonetheless, I called Kuhn and obtained some of his protein, and sure enough, in short order, Jeff Benevik was able to show that when added to our reconstituted system, this visual arrestin was able to restore the ability of GRK2 to desensitize the beta-adrenergic receptor, granted at very high concentrations. Then, shortly thereafter, Shinohara at the NIH cloned the cDNA for retinal arrestin. reasoning that what we were losing in our purification might be not only functionally analogous with the arrestin molecule, but structurally homologous with it, we obtained his clone and performed low stringency screening. And Martin Losi, then a fellow in the laboratory, was able to pull out a cDNA for a new molecule, which was 70% sequence identical with visual arrestin. We called it beta-arrestin. Within another year or two, another fellow, Hoveder Tramadol, pulled out another molecule very similar. We called it beta-arrestin 2, renaming the first molecule beta-arrestin 1. Now, with authentic, validated, recombinant materials in hand, we were able to set up parallel reconstitution systems with either rhodopsin kinase phosphorylated rhodopsin or BARC, GRK2 phosphorylated beta receptor, and show that for the visual system, the arrestin or visual arrestin was very potent in turning off signaling, whereas the beta arrestins were rather weak. Conversely, for the beta receptor, arrestins 1 and 2 were very potent, and visual arrestin was very weak. So these results then showed that there was conservation of the turnoff mechanism, not just the signaling mechanism, which we knew about, but there was conservation of the turnoff mechanism, desensitization across the wide family, from rhodopsin to the beta receptor, however, with great specificity for the kinases and the arrestins involved. Today we know there are 4 members of this arrestin family. 2 are present only in the retina. Beta-arrestins 1 and 2 are ubiquitously expressed. There are crystal structures of all members of the family. They share the same fold, 2 domains, consisting almost entirely of anti-parallel beta sheets, connected by a flexible hinge region and stabilized by a polar core. So where this brought us to by the mid-90s is as shown in this cartoon, illustrating that there are 2 universally applicable paradigms which govern the function of essentially all GPCRs. These are activation and desensitization, and are in turn based on the understanding that 3 families of proteins, and only 3, share the remarkable property of being able to interact in essentially universally in a stimulus-dependent way with the GPCRs. And these families, of course, are the heterotrimeric G proteins, the Gproteincoupled receptor kinases, and the beta-arrestins. When the receptors are activated, they interact with the G protein, leading, of course, to the well-known cascade of enzyme activations and phosphorylations, which lead to second messenger generation and signaling. However, the activated receptors are phosphorylated by GRKs, generally on multiple serines and threonine residues. This facilitates the binding of these beta-arrestin molecules, which bind to the receptor and then sterically interdict further signaling by the G proteins. This leads to receptor desensitization. But over the past decade or so, there's been a dramatic change or evolution in this understanding, as we've come to realize that this GRK beta-arrestin system is actually multifunctional. At the very same time that it desensitizes the receptors, it also sets up independent signaling activity of its own, by virtue of the fact that the beta-arrestins can serve as scaffolds or adapters to organize various signaling elements. And so the GRK beta-arrestin system is itself a signal transduction system, leading to activation of a variety of signaling pathways and a variety of cellular consequences. I think the MAP kinase systems, especially ERK MAP kinase, have been the most extensively studied. In addition, the beta-arrestins bind to various elements of the clathrin-coated pit machinery and thereby serve as adapters and mediators of clathrin-mediated endocytosis. So, the beta-arrestins serve 3 essential functions. They desensitize G protein signaling, they lead to receptor endocytosis, and they lead to receptor signaling. Now, in the course of studying these new signaling activities of the beta-arrestins, we made an interesting discovery just a few years back, and that's shown here. That discovery is of what we call biased agonists. So a biased agonist is a ligand which stabilizes a particular active conformation of a receptor, thus stimulating some responses but not others. 7 transmembrane receptor ligands, for example, can be biased toward a particular G protein or beta-arrestin. Mutated receptors can also be biased. By way of illustration, let me show you some simple cellular data which compares the ability of 3 different ligands for the angiotensin receptor, another GPCR, to promote interaction of the receptor with either G protein or beta-arrestin. Before I do that, though, let me just give you one further point about this, which is that the existence of biased agonists calls into question, in a sense, one of our most fundamental assumptions of receptor theory, and that is the classical 2-state model of receptor action. So in the classical model, when an agonist combines with the receptor, it promotes the formation of an agonist-receptor complex with the R-star form representing the active state of the receptor. In this simple model, virtually all signaling is a function simply of the concentration of AR-star. But the existence of biased agonists, which can promote one form of signaling, say G protein, but not Berestin, is formally incompatible with that. And so it basically calls for the existence of multiple active conformations of the receptor, shown here for simplicity as 2, one being able to activate G protein and one being able to activate Berestin. So as I said, I want to illustrate this whole idea of biased agonism with these 3 different ligands. Okay, and we're going to look at their ability to promote interaction of the receptor with G protein or beta-arrestin. Okay, so in these curves, these are classical dose-response curves. Shown in red is the ability of an agonist, in this case, for example, angiotensin, to promote G protein signaling, measured as turnover of phosphatidyl inositol. Classical second messenger assay. And here shown in black, we're looking at the ability of the ligand to promote beta-arrestin recruitment from the cytosol to the plasma membrane. And you can see with angiotensin, which is an unbiased or balanced ligand, that both of these curves are very similar. If instead of using angiotensin, we use a so-called ARB, angiotensin receptor blocker, valsartan. This is a very commonly used drug in clinical medicine, cardiovascular medicine. You can see that because it's an antagonist, it doesn't stimulate either process. But look at this last one. This is a compound, let's call it TRV, Trevena 027. This is a Berestin-biased ligand. It is, like angiotensin, which is a naturally occurring ligand, this is a synthetic octapeptide analog of angiotensin. And as you can see shown in red, like valsartan, it's an angiotensin receptor blocker in the sense that it has absolutely no ability to stimulate G proteins. In fact, it blocks G protein activation. Nonetheless, it's quite robust in its ability to activate beta-arrestins. So this is a Berestin-biased ligand. Now, as I mentioned before, one consequence of Berestin recruitment to the receptor is to promote signaling, okay? But we were interested in gaining a more global understanding of the various kinds of signaling that might be induced. And so we took cells that were expressing the angiotensin receptor, and Kevin Zhao in my lab conducted a mass spec proteomics analysis in which we stimulated these angiotensin receptors with our biased angiotensin ligand. So remember, this is a ligand. It can't activate G proteins at all, but it does activate beta-arrestins. We then used mass spectrometry to determine all phosphorylation reactions which occurred in the cell when we stimulated signaling only through Berestin and without G proteins. And I won't take you through this in any detail, but we discovered all manner of signaling: MAP kinase signaling, PI3 kinase AKT, DNA repair mechanisms, cell cycle and development pathways, all kinds of cytoskeletal reorganization pathways. The only take-home message for you here is that signaling downstream of Berestins, which is often going on in parallel with and independent of G protein activation, is very, very diverse. It's extensive. Many of the pathways activated overlap with those activated through classical G protein signaling. But almost invariably, even when the same pathways are being activated as G proteins, they lead to different cellular consequences because the activation is going on in different regions of the cell. Now, we believe that these findings have potentially important consequences or implications for the development of new therapeutics. Let me show you why. Again, using the angiotensin receptor as an example. I showed you that when you activate a typical GPCR, such as the angiotensin receptor, you activate G proteins and berestins. But in some circumstances, G protein activation through angiotensin can have deleterious effects. For example, we know it raises blood pressure. Angiotensin is the most potent vasoconstrictor we have in our bodies. That's okay, but not necessarily if you're hypertensive or have cardiovascular disease. Berestin signaling, on the other hand, seems to have some cellular effects which might be beneficial. For example, it's apoptotic and cytoprotective. Angiotensin receptor blockers block these potentially deleterious effects that are mediated through G proteins, hence the fact that they're amongst the most widely used drugs in the world for the treatment of heart disease and hypertension. But of course, they also equally well block the potentially beneficial effects that go through beta-arrestin. So our hypothesis is that a biased ligand, such as that 027 compound I showed you, which, like valsartan, completely blocks deleterious G protein effects, but which, unlike classical ARBs, actually promotes berestin-mediated signaling, might have some added therapeutic benefit. And in fact, that compound, 027, has been shown to slow progression of heart failure in animal models, to lower blood pressure, to increase cardiac performance, and to have anti-apoptotic effects which are not present in classical angiotensin receptor blockers. And in fact, this compound is currently in phase 2 clinical trials for treatment of acute congestive heart failure. Now, I wouldn't want to leave you with the idea that only berestin-biased ligands might have therapeutic benefit. It's equally, we can equally imagine that under some circumstances, G protein biased ligands might have therapeutic benefit. Let me give you one example of that. Consider opiates, like morphine. These are by far the most efficacious agents we have for the treatment of severe pain. Their pain-relieving effects are mediated through a typical GPCR, the mu opioid receptor, signaling through GI. But we also know that opiates have all sorts of difficult side effects. They cause severe constipation in some patients, they cause respiratory depression, and they lead to tolerance, which is desensitization, in which greater and greater doses of the drug are necessary. All of those unwanted side effects have been shown to be mediated through berestin-2 signaling, and in berestin-2 knockout mice are completely lost. So this suggests that if you had a ligand for the mu opioid receptor, which was completely G-biased, you should have all the pain-relieving effects, and you should have reduced or loss of all those berestin-mediated side effects. And just such a compound is in early-phase clinical trials. I've given you just 2 examples, but hopefully from that you can see how our new understanding of these parallel signaling mechanisms of G proteins and berestins, understanding that was gained initially at the biochemical level, may well be leveraged and translated into new therapeutic approaches. These are the people that I owe my Nobel Prize to, the fellows who've trained with me over the years. I've trained over 200 students and fellows. This picture was taken, it's one of my favorites, I think you'll see for obvious reasons, 10 years ago at the time of my 60th birthday, when about 100 of them returned from all over the world to help me celebrate. And I just want to point out one individual who was hidden away here in the last row. That's Brian Kobilka, who shared the prize with me this year. Thank you very much.
Dear Professor Lefkowitz, Thank you for a fantastic and inspiring lecture. Now, dear audience, it's actually you who are going to be activated. There is microphones out in the audience, so please take a chance to ask a question to the Nobel Laureate of 2012 year's Chemistry Prize. Anybody?
It doesn't have to be about the lecture either.
Anything in life, basically.
Yeah, I can hear.
I'm going to share it now. So, I'm not ask a question, but just have to make a comment because some of you know that I worked for years, 1981, 1982. But for those who doesn't know what heroic work it was to isolate and purify these receptors, I would just make a comment that I remember in the lab that you used the southern grass frog. And the laboratory piled up with these frogs in masses and out in the corridor. And I don't know how many, maybe 10,000 of frogs or something was used to get the red blood cells for this purification. Maybe you can comment so that people really understand what heroic work was involved in these experimentations.
So I'm not sure, I had trouble hearing the question at the end. I mean,
Yeah. So the question, I think, if I understand it, is maybe comment a bit on the great effort you made when you tried to purify first receptor.
yes, this was really a remarkable business. All the initial purification work was done using frog erythrocytes. Now you might say, why frog erythrocytes? Well, it turned out from various screenings we had done, they had a uniquely high concentration of beta receptor. Now, when I say uniquely high, it's still probably 100th or 1,000th of what we can get today with overexpression of cloned receptors. But at the time, it was the best we could do. So, you know, you can only get a couple of mils of blood out of a frog. So we used to take delivery of 2,000 frogs, 2,000 grass frogs, Ranapippias, to the lab. And the entire lab would come together, even those who were not working on this project, purification project. And everybody would work for like 2 days straight, just getting the blood out of these frogs. That was just the beginning of the process. And, you know, Durham, North Carolina, where I live, is very hot, especially during the summer. And some of our shipments, many of the animals would be dead from the heat when they arrive, and that makes an awful bad smell. And so there were a lot of problems associated with that. The alpha-2 receptors we purified from outdated human platelets, so that was at least a little easier to do. We could get large amounts of material. The alpha-1 adrenergic receptors were purified from a cultured cell line. And at the time, we used to grow 100 liters of these cells a week. Again, a heroic effort. But just to give you an example of what was going on. So one particular fellow was in charge of that project, purifying the alpha-1 adrenergic receptor. She worked for almost a year, every week, growing 100 liters of cells and purifying less than a microgram of receptor ultimately with these techniques. At the end of a year, she had after a year, somewhere between 25 and 50 micrograms of receptor protein. We cleaved it with cyanogen bromide, and now the cyanogen bromide fragment mixture was put onto an HPLC column so we could isolate the peptides to try to do microsequencing. We knew that with that little protein, there was some possibility that once having injected those peptides on the column, we would never see them again. Any of you who've done column chromatography of tiny quantities of peptides knows that that's always a risk when you have very little material. And I still remember the fellow, she was so nervous, remember, she had invested a year of work in this, that she asked me to stand with her as she injected it onto the column. I mean, I've convinced my people that I have very good luck and that somehow I will bring this to them. And I still remember her hand was shaking as she injected the mixture onto the HPLC column. Fortunately, some peptides came off, and they were, in fact, alpha-1 adrenergic receptor peptides. So this just emphasizes this was a very fraught effort and went on over 10 years, and it was very, very difficult. But it was the key to the whole business, because once we had the first few receptors cloned, then you never had to purify your receptor anymore. You could take the sequences that were there, and by low stringency screening of enriched cDNA libraries, you could bring it out. But sometimes you just have to do that very difficult work.
Thank you. More questions? Yes. There. Agneta first. Yeah.
Thank you for a wonderful lecture. When these receptors are activated, they also activate other receptors through the transactivation. Can you comment a bit about the mechanism? Is that through activation of the G proteins or the beta-arrestin? And can we use for regulating this also this type of bias?
Again, I missed the very last part of this. Could you
Agneta, could you repeat it?
just the last part of the question.
I was just wondering about the mechanism for the transactivation. Is it through the activation of the beta-arrestin and the SARC kinases and so on? And do you think that we can use this type of biased pathways to regulate this other type of activations?
So I think the question relates to an interesting phenomenon which has been described in recent years called transactivation. And this is a fascinating process. You know, a number of years ago, if you went to a meeting on Gproteincoupled receptor signaling, you never heard anything about tyrosine kinase receptors, and vice versa. But today we know that when you activate some GPCRs, they transactivate certain receptor tyrosine kinases, like the EGF receptor or the insulin-like growth factor receptor, and vice versa. Turns out that there are a number of different mechanisms by which GPCRs can transactivate these receptor tyrosine kinases, like EGFR. Usually it's through a process where activation of the Gproteincoupled receptor leads to cleavage of certain molecules, certain precursors in the cell membrane, and those cleavages lead to release of, for example, ligands for the EGF receptor. Initially, those processes were thought to be exclusively G-protein mediated. Today, we know that there are also mechanisms through which beta-arrestins can mediate such transactivations. In particular, one pathway, which has been shown by several laboratories, involves a berestin-mediated activation of SARC kinases, and then those SARC family kinases activate these cleavage enzymes, these proteases, which clip off the EGF receptor ligands. So once again, it's an example of where a certain end result, transactivation of a growth factor receptor, can occur in either a G protein or a berestin-mediated mechanism. Yeah, you.
Hello. It was quite impressive, like this purification. How it took you a decade, right? But I was wondering how you actually motivate your students, squeezing blood out of frogs, and really growing cells for a year, and how do you motivate yourself for a decade on one project?
Yes,
which is quite
this is a very good question, and this gentleman here is one who would also speak to that, because those of you who have read about our Nobel Prize know that what Brian has accomplished in recent years is the obtaining of high-resolution crystal structures of these receptors. And while admittedly working on other projects, he was trying to crystallize the receptors for 15 years. And it was only like at about year 12 or 13 that he finally succeeded. And many other people were trying to crystallize the receptors. Brian is, as you know, not a crystallographer by any stretch of the imagination, although he was collaborating with crystallographers. But many, what I'll call hardcore structural biologists, were competing with him, and they did not succeed. So I think it does require a special level of confidence or vision or craziness, maybe. I don't know. And maybe we were deluded because it might have turned out very differently. But I guess you really have to believe in what you're doing and believe that it's an important goal. And I think, one of the important things about being a good mentor is to be able to somehow make your trainees believe that what they're working on is important. And of course, the only way to do that is to believe it yourself. I remember at this celebration 10 years ago, there were several symposia held. One of them was a symposium at which only my former trainees spoke. And as each one got up, they told a little vignette about what it was like to work with me. And one guy, I'll never forget his story. This was Rick Serione, who was the guy who did those reconstitution experiments many years ago. And he told how when he was working on his project, and whenever he would get down and he would come talk to me because things weren't going well, he would say he would always come away, he was telling one of his colleagues in life, he'd always come away with the impression and the belief that his project was far and away the most important project in the laboratory. And that I, the mentor, believed that his project was far and away the most important one to me. And then his colleague in the lab said, wait a minute, that's what I thought about my project. And then another guy said, well, wait a minute, I thought my project was the most important one to Bob. So somehow I had convinced everybody in the lab that their project was most important. And you know what? I believed it. So I think, so I think that's a big part of it.
By that, I think we close the session. And once again, thank you so much for visiting Uppsala University and us in Uppsala. Thank you.